Hi, Frank. Hi, everyone. Inviting you to speak. How are you, Frank? Nice seeing you here. Hi, Bet. How?
Hey, Katerina. Yeah. This will be a very interesting talk, huh? So I just did a Google. I think Boyden it confirms my memory that he's one of the I think he was a graduate student in Nagel's lab. That was what Wikipedia says. When the optogenetics tools, they were one of the group that first confirmed. Oh,
Yep.
Serena here. Hi, Serena.
Hello. Couldn't miss this one.
Yeah, he's a major technology developmental person that brought a lot of great tools for neuroscience that are really important and broadly used nowadays. And yeah, I put the link up. He has various websites. He has his own website, then from his group, synthetic neurobiology group website, and then he also has the MIT Media Lab, his website there, and the McGovern Institute at MIT. So I don't know. I might switch around. Hi, Victoria. How are you?
Hey there.
Hello. Wow, happy to see this crew.
Hi.
Yeah,
Right?
it's so cool.
Yeah, how is everybody?
Busy. I slipped into a 7-day-a-week work pattern.
Okay. Yeah, I know about that.
It's been going okay, though.
Yeah, good. I hope you're getting some self-care time.
I'm retroactively calling it that. Selectively. Sure.
Do we have to come over there?
Yes. Yes, you do. Come on over. Enjoy the sunset with me.
Oh, that sounds great. It's pouring here. It's been pouring all day, and yesterday it snowed all day, which was pretty fun.
Oh, no.
Yeah, well, the snow was great because I just expected it wasn't even predicted. It was a kind of a surprise.
I don't want to ever see the stuff again. I have latitude restrictions. Too much time in New York.
It is really distracting.
Well, the part about it being so cold doesn't help.
Oh, yeah, that part. I just put on a lot of layers and go outside in it. I just will never get used to it. It's just so pretty and quiet. It's so quiet. And then you just look up into it as it's falling,
Mm-hmm.
and you can see, it's just this dimensions of just snow. And it goes forever. I just can't get used to that sight. But I hear you about the cold part. It's that we get it so infrequently. It doesn't usually stick for more than a week if it does, so it is a novelty.
Yeah, I guess it's different when you can't escape it. That'll take a toll on you. What do you think about snow, Katerina? You're still there.
Okay, sorry, I couldn't come back to without finishing that. I love snow. Well, I love skiing. I love snow. As long as it's not too cold, I love it. We used to go in Switzerland and spring break. Higher up, you can be basically in a t-shirt, and because the mountain is still so cold at night, and also the ground, you can do skiing in a t-shirt. It's amazing. You have the view. It's silent. Snow makes everything silent. I love it. You know,
That
I love
is
different things. You
beautiful.
know,
Yeah,
not the
that is
whole
really
year,
cool.
but yeah. Sorry, Victoria. Go ahead.
Oh no, I was cutting you off. Cutting off. Yeah, just the part I forgot about the part about the skiing and the t-shirt. That was great. To be able to be in that in snow and not have a million layers on, that's a kind of freedom.
Yeah, I agree. It's wonderful. Yeah, I like, different things throughout the year. Just summer or just fall or just, that would be I like a mix. However, I get sick of New York winter by like February, then then it's enough. And I could be done with it, but that's not how it works. So, like, I don't like March. February, March is it gets old then. Let's say it that way. And welcome everyone. We'll start on top of the hour. In the meantime, there are some resources pinned on top of the room, but also even more in the chat. Feel free to check them out in the meantime.
Does anyone up here make roasted pumpkin seeds, or just the seeds? Cook the seeds of your winter squash? Hey, Denise.
Hi everyone, I do. And also, don't eat yellow snow. That's my snow contribution.
Ew. Well,
Ew.
I'm just asking because I discovered that you don't have to separate them from all of the pulp. You can just kind of, I mean, you remove the bigger parts, but you can just, it just saves so much time to just put them in the oven
And
with
just let it
some,
roast off the web. Yeah.
yeah, with a little, I put on some olive oil, but yeah, it just kind of cooks or it tastes sweet. It's fun. It used to just take so much time and then you get all over your fingers. And anyway, that's my cooking tip for today. Thank you.
You can also use a blender, one of those hand blenders, if you want to get that stuff out. I haven't done it myself, but I saw a video. It seemed pretty convincing.
I'm sorry, how?
It's kind of like a stand mixer, but the handheld ones. Imagine an egg beater, but it's mechanized. And I know probably not everybody has one of those. I definitely don't have one, but
Oh, right.
Yeah, I just tried my hand at these blenders that you mentioned. First cake that I tried. The ice cream cakes with the blender, the egg white is much better. It's like change the into another form of material. I would say it's like phase transition. It's very interesting.
Well, there's some sorts of cakes you don't want to use the machine because then you overbeat it. It's supposed to be more clumsy or clumpy. So sometimes it ruins it.
Yeah, you don't want to
See
develop.
that's the difference. Newbie. I just beat it to the death. It's become completely like foamy and stuff. I thought I was wondering, why. I mean, it turns out the ice cream cake in the end was okay, but it's just somehow a little bit icy. It's not as creamy, so maybe that's the reason.
Well, if it's a glutinous flour and you're making something like muffins or pancakes, you don't want to overbeat. You want to keep the clumps, as Katarina was saying. And if it's cake, then often it's a cake flour, which isn't such a high gluten flour. And so you want to. I don't really make that many cakes, but I know you want to beat the hell out of it, or you can.
Well, this should be interesting. Did Professor Boyden confirm, or?
Do you think I would keep the room up if you didn't confirm?
Well, I'm just dropping the subtle hint.
Well, if you don't have time to wait a few minutes,
I have time, Katerina.
Yeah, I emailed him 5 minutes ago, with a link, but he confirmed the last time October 31st. We talked about if we should do slides or not, and stuff. So yeah, should be here. Hope.
Okay.
And yeah, in the meantime, we can talk a little bit about the room, what this is about, and yeah, make this waiting time a little bit interesting. So, as I said, at Boyden, he is at MIT mostly. He has the synthetic neurobiology group. I posted the link on top of the room. Check it out. It has really a lot of interesting, resources, including, who with the latest Nature news in nanotechnology. Where I don't know if you learned about this, but there's Ed. Hi, Ed.
Hello, can you hear me?
Yes, perfect.
Hello.
Great. Cool. I just want to I haven't used Clubhouse in a couple of years, so I'm trying to make sure I'm doing it correctly.
Yeah, you're perfect. And
Wonderful.
welcome back. Thank you for coming back after 2 years for us.
Yeah,
Honor.
great to be here. Thanks so much for having me.
Yeah, that's wonderful. And yeah, let's start maybe with an introduction. I think everyone knows you, but maybe in case somebody is here that doesn't know anything about neuroscience, I'll give the audience a short introduction, and then we'll start a conversation, I think. Is that good?
Yes. My name is Ed
Perfect.
Boyden. Good. Yeah, so I direct a group at MIT at the McGovern Institute, which works on technologies for mapping and controlling the brain. Many of these tools are in widespread use in neuroscience, but also increasingly in many other fields as well, from cancer biology to studying COVID-19 to trying to confront aging. And so our hope is to really get biology down to what you might call a ground truth level of description, the fundamental building blocks of life and how they interact and work together, both so we can eventually, hopefully, have a theory of life. Can we simulate life in a computer even? And hopefully also be able to cure diseases which are currently intractable. So that's kind of the bird's eye view of what we're trying to do.
Yeah, that's really a wonderful introduction, and it's really interesting work you do. And I posted on top of the room a link to, you have different websites. One of the websites that so if people want to check it out, additionally after the room, check out different papers, resources. Please click on the link. It's also shared in the chat in case we switch to something else. And yeah, I'll just start with like a few questions that are kind of just pointing to different works you did. So usually Victoria starts with really interesting general questions, if that's okay, and then I'll take a turn. Is that good?
Wonderful.
Okay, Victoria, go ahead.
Here I am. I just have to get this peanut butter off my hands. Okay.
Wonderful.
Yeah. Um, sorry, it's all about the timing. Yeah, so
Absolutely.
welcome. Welcome, and it's fantastic to have you back. And Science Society is really appreciative of you coming and sharing your time with us. So just my question for you is to give us more background about you to lead us into the body of your discussion. So if you can think back through your life to where you noticed that you felt an affinity towards science or scientific thought or research, and this could be in your childhood or any time, just when did that spark hit you?
Yeah, great question. Well, I grew up in North Texas, in a suburb of Dallas, And when I was young, I quickly became obsessed with, what is the meaning of life? I went through a religious phase. My parents would drop me off at church um, around 8 years old. And around age 9, I started wondering, can we understand all of this more directly? Can we understand life? Can we understand existence? And I became very philosophical. And a couple years later, around age 12, I went to the science fair in Austin, Texas, the capital, and people were doing real science. They were looking at cancer genes. They were studying, at universities, and I just became really obsessed with doing science. And so I became really ambitious. I really started studying hard, and I ended up leaving home at age 14 and going to a special program called TAMS, the Texas Academy of Math and Science, where they take a bunch of high school students across Texas and have them basically go directly to college, skipping their remaining grades. And that was wonderful. I worked in a group that was working on the origins of life, Paul Braidman's group. They were trying to create DNA out of clay, which didn't work, or you would have heard about it, but it was still a fantastic training ground to think about life and to do science at the border of philosophy and practicality. That's how I got started.
That sounds just so wonderful. I'm imagining how that was to be 14 and be taken so seriously and have your abilities and your curiosity taken so seriously that you were given all of this, these challenges. It's wonderful. And
Yeah, yeah.
yeah, so can you take us maybe step by step through events that brought you up to your current research today? I'm sure I'm just like they're myriad, they're infinite. But
Sure.
key points. Thank you so much. It's very. I'm just really. It leaves so much to imagine. We could just talk about that. What that experience was for a kid to go away and your thought processes and what you were wondering about. But anyway, I've given you a question. That will be for another day. Thank you so much, Ed.
Yeah. Well, so in summary, after that, I really stuck with projects that were at the border of philosophy and science. I transferred to MIT, where I switched my major from chemistry to physics and ended up studying engineering as well. I was an undergraduate for 6 years, and I ended up working on quantum computing for my master's degree. Again, sort of the border of the strange and the scientific and the practical. And I learned something, working the origins of life, that's a really hard problem. Working on quantum computing, another really hard problem. And third time's a charm. In 1998, I got really interested in neuroscience um, and the brain. I thought maybe we could just try to solve it in terms of the underlying chemistry and physics. It's a less fundamentally difficult problem than, let's say, quantum computing or the origins of life, which are sort of very fundamental. And yeah, it turns out that all that chemistry experience and physics experience prepared me to take a novel approach to neuroscience. So yeah, in 1999, I switched into the Stanford PhD program and studied learning and memory with Jennifer Raymond and Dick Chen. And then as a side project, did this project on controlling the brain with light in parallel to my day job of the PhD, and that became optogenetics, which became a big hit. And that's led to my group at MIT.
Thank you. And I'm again just thinking about that as giving you, since you had those tools to further your research and questioning at such a young age, I'm wondering how that influenced you as well to, just to pursue your own original thought.
Yeah. Well, two things happened. One was because I started college so young, and as 4 years younger than people, I always felt like I had some extra time to bet on big, risky things. And then I was lucky, so things worked out really well. And so then I would try even bigger, riskier things, and that kind of kept going to the current day. And yeah, so but also coming from a different field, right? Training in chemistry and physics and engineering was very helpful because um, I think I came to the field with maybe a different set. I was going to say less preconceived notions, but maybe a better phraseology is different preconceived notions about the brain. And one of them, of course, was that we should use a technological approach to confront the brain. Um, we need to build the tools so we can see what's in the brain, right? What we don't see, we cannot understand, and to control the brain so we can fix it. And that's now led to technologies that are showing promise in human trials for blindness and Alzheimer's and other conditions.
Thank you. Thank you for that detailed description. At this point, I'm going to just pass the mic to you. And you know, you've been here before, but we're here to moderate the room for you.
Thank you.
Afterward, if you'd like to have Q&A, then we can do that. If you prefer to have questions driving,
Oh,
your talk along.
Vic-
Oh, sorry,
Victoria,
sorry.
we are just doing a
Ah,
discussion mode
take it away. Yeah,
style, not
take it away, Catherine.
like classical. Oh,
Yeah,
yeah, thank you.
take the mic.
Um,
I'm done. Thank
thank you. That's really wonderful that you had this opportunity to take risks. Um, I think that's really a key. Do you think that's a key for developing like this groundbreaking type of technology? Because it is like, just for everyone that doesn't know, like optogenetics and these technologies that developed with his colleagues are like everyone in neuroscience basically is using it to do precise to dissect out precisely what is happening in the brain and then hopefully in the future also treatment. So yeah, do you think to have this ability to do high risk is really crucial to come up with progress like this?
Well, the funny thing is, if you look at optogenetics or expansion microscopy or other tools you develop, it would take us years to kind of converge in the right approach. But then once we started the experiments, it would converge pretty fast. So I think there's a way to reduce risk. And a lot of the things we do look high risk, but because we practice a certain way of confronting problems, I actually think risk can be reduced. Um, and so many of my group's alumni are confronting climate change or trying to revolutionize education or trying to revolutionize 3D printing or other areas. And I think it's because there is a scientific method which might be optimized for trying to be very revolutionary about a topic. And um, yeah, so I think there's a way of reducing risk that could be thought more about. Um, I never have time to do it, but I thought about writing a book on this topic. And I started it 7 or 8 times, but it does seem like you can think about a problem very systematically. Try to think of every possible solution to the problem, for example. Um, design strategies that reduce risk by what I call constructively failing. You try something and it fails, but it teaches you what to do next because the failure shows you something the world's never seen before. Anyway, I think there's many methodologies you can invent that can directly convert risk into low risk or failure into success. And so that's something that I think a lot about.
Yeah, that's really interesting. I would love to read that book, so please,
Hopefully I'll
please.
plan to work on it someday.
Yeah, that's always what we say, right? One day we will have time for it.
Yes, one of these decades. One of these
Yeah.
decades.
I hope you have. And um, another thing I wanted to point out is the expansion microscopy, which is, I think, also a wonderful project that you did, not just because of the elegancy and how useful it is, but also it enables a lot of labs that usually wouldn't have money to go into a more granular data, like smaller data, and to visualize it. It kind of democratizes this type of field. So how did you start working on this project? It's really interesting.
Well, with actually my very first postdoc, Brian Chow, we were brainstorming how could we separate all the molecules in a sample so that you could make room to tag them. And then you could also do nanoimaging. This is in 2007 or so. But it turns out that, super resolution microscopy had come out the year before, and we got busy with other things. Um, and then in 2012, Fei Chen and Paul Tilburg were working in my group. Fei was doing storm microscopy, and Paul was doing electron microscopy. And we were trying to do nanoimaging, and it was hard. So we started working, thinking, wow, if we could expand things, that could be really useful. And that led us down to the passive expansion, and we started pursuing it, I guess, around summer 2012, I guess. And then by early 2013, it was showing signs of working. And so for those who don't know what it is, we take a specimen, a preserved specimen of the brain, and we can form a dense spiderweb-like mesh of baby diaper polymer inside the brain, inside the cells, around the cells, just permeating it. And if you do it just right and add water, the baby diaper polymer swells, but it'll make the brain physically bigger. So yeah, now anybody can do nanoimaging. The fundamental building blocks of life are nanoscale, and now, so you want to map the brain, you got to see those nanoscale things. Expansion lets you do that. And so, yeah, that's over 400, 450 results have already come out doing expansion. Probably over 1,000 groups are practicing it. It lets anybody image down to the fundamental resolution of life.
Yeah, I'll just make a comment and then I'll pass it on to everyone else because I think everyone is excited to talk with you. So, yeah, I think it's one thing to do a work to dissect out the path where we're doing like tiny increases of knowledge, but giving people tools that even make their work affordable is really a whole nother level. So yeah, thank you so much for doing that, and I'll pass the mic to Frank. Thank you.
Oh, hi. Thanks, Katrina. Thanks, Dr. Boyden. Your work, groundbreaking work on optogenetics and it cannot, be, said more that it's very, make so much impact on in the community of researcher. I myself consider myself an engineer working with plastics. So what Katerina was just give an introduction on the your new invention, new work that the expansion of polymers. So I was wondering what type of a polymer and how much it expands. I actually myself has a self-healing type with the very it's we call it um, it's a trim it's a it's type of now it's been a while, but it's a 3D printing material that were made out of a trimer, the PE and PP, so that it's actually can under stress it can kind of has a maximum 5 times of extension. So by that we propose it can be used. It's just using regular light. It shows a very regular pattern of types of absorption, and so that it, because of the large expansion, it can function. We propose it can function as a kind of a magnify, but that's a very macro scale of improvement. So you're at the nanoscale is very groundbreaking. So I was wondering, what type of material and how much it extends.
In our first paper in 2015, when we announced the discovery that we could magnify brains, we used sodium polyacrylate. So it's a free radical synthesized hydrogel. The physics of studying this goes back to the 1970s, 1980s. There's a paper by Toyoshi Tanaka called Phase Transitions in Ionic Gels, which describes the phase transition-like physics of the expansion process. And that's the polymer we used. You can create it several different ways. People have made different flavors. So our initial flavor expands 4.5-fold in each direction, about 100 times in volume. Several groups now have made versions that expand 10 times in each direction, 1,000 times in volume. But in 2017, we showed that we could take a specimen, polymerize and expand it. Then we can form a second polymer in the space opened up by the first expansion and expand it again. So you can do that over and over again if you want to. And so, yeah, I mean, many people now are using all sorts of different expansion methods, different polymers to expand. I mean, in fact, some people try to do it even without polymers. They just add a bunch of chemicals to the specimen. The specimen absorbs those liquids full of different salts and different, materials, and then it expands just by swelling.
Yeah, that's amazing. I think the method of delivering is also. So I assume this is. I haven't read your paper yet, so I'm quite sure. I mean, it's been done like in vivo, right?
Oh yeah, over 1,000 research groups are already doing expansion microscopy, and 400, 450. Let me see. I keep a counter here. How many papers? 454 experimental results have put on bioRxiv so far. So rapidly spreading. Um, it's very easy to do.
I see. I'll go
In fact, I
read
just got a
the. Thank
Google
you.
alert that today 4 more papers came out doing expansion. So it's spreading very, very quickly.
Thank you. Yeah. Great. I'll pass the mic to Serena.
Well, so hello. There's so many aspects of your work that are interesting. My background is in chemistry, but that was in the 90s, and I sort of had a walkabout throughout different topics. I did do some work with channel rhodopsin, or not, sorry, rhodopsin actually, simulation work, in diving into the mechanism of rhodopsinism in the visual protein. I'm less read on the actual detailed mechanisms of optogenetics. I'd be interested in hearing a little bit about that, but I also want to comment that the expansion microscopy aspect, really brilliant. Amusing how you introduced it as a baby diaper polymer. I'm curious if the actual original thought that sent you down that road into exploring different polymers and their properties of expanding, for example, how that came about. If we have time later and coming back, I'll open up the origin of life can of worms. But let's let other people ask first.
Great.
Curious, yeah, the
Go ahead.
original idea of expansion, and maybe some comments on the mechanisms of optogenetics.
Great. Let's start with the optogenetics. So rhodopsins are the molecules in our eyes that convert light into downstream chemical signals. It turns out microbes all over the tree of life, all over the earth, contain proteins that convert light directly into electricity. They look like the rhodopsins in our eyes at a gross level, but they contain ion pumps or ion channels instead. So in optogenetics, what we discovered was that we could take the genes that encode for these light activated proteins, we can express those genes in brain cells, and they work. They're safe enough, they're fast enough, they're powerful enough that you can shine light on a brain cell expressing this protein, and you can turn that brain cell on or off. So that's being used by thousands and thousands of neuroscientists to activate brain cells with light, express the gene, shine the light on the cell, and you can turn a brain cell on, electrically speaking. By turning a brain cell on, you can figure out what it can initiate or sustain, a behavior, a pathology state, a therapeutic state. By turning a brain cell off, you can find out what it's needed for. Delete it and see if it's needed for a behavior or a pathology state or a therapeutic state. So that's optogenetics in a nutshell.
Just a
Go ahead,
quick follow up.
jump
Now,
in, everybody.
is there a cofactor involved in that mechanism that needs to be externally regenerated, like in rhodopsin, or is that just cleanly reversible?
So there is a cofactor called all-trans-retinal, but
Okay, that's
amazingly, it seems to be spontaneously present in the brain, which is amazing. Sheer dumb luck there. And amazingly, it doesn't need any extra machinery to generate it. It's a closed photocycle for the aficionados.
I see. So there's no cis-trans isomerization in the activation, like with rhodopsin.
Oh, there is. It summarizes absolutely, but it goes back to the initial state spontaneously.
Huh. Well, that's convenient.
Absolutely. Isn't it? It's just all biology, all the great stuff is luck, right? CRISPR was discovered, in yogurt, right? Green fluorescent protein found in jellyfish. None of this stuff had to work. None of this stuff had to be there, right? All the great treasure is hiding in plain sight. You just have to go find it.
Very cool. And so the origin of the idea for expansion.
Yeah, so as I mentioned earlier, Brian Chow, my first postdoc, and I were brainstorming how could we physically magnify an object, and we started looking at some hydrogels that would expand a bit by twofold or so. But super resolution microscopy had just come out the year before, so we thought, a lot of people are doing nanoimaging. And then two then grad students, Faye Chen and Paul Tilburg, joined my group. Faye spent his first year in my group doing super resolution microscopy, and Paul spent his first year doing electron microscopy. Turns out doing nanoimaging is really hard, and so we started revisiting expansion and found some papers on polymers expand more and gave it a try.
Okay, thank you. I'll pass the
You're
mic to
welcome.
Denise.
Thanks, Serena. Wow, so many interesting things we covered already. I was going to ask a little bit more about so there have been several papers that are submitted, and this is mostly stuff on a lab probably or a bench. I'm curious what you see in terms of clinical setting, or where do you see these things, or the fruits of your research? How do you see that translating to therapeutics, or to be able to benefit patients? Because I can hear it from the way that you speak that that's one of the things that you'd like to see happen.
Well, it is happening, yeah. So a European team has taken one of our optogenetic molecules, and last summer they announced that they could help a untreatable blindness patient to have a partial restoration of functional vision. Um, a group at MIT, Li Wei Sai's group, used optogenetics to discover a brainwave that might help clean up Alzheimer's disease. And we've now been helping her um, to see if you could induce those brainwaves with movies. And then, imagine a movie that could help treat Alzheimer's disease. The expansion method, if you early in a disease, the changes are small, right? That's almost the definition of early. And we and others have shown in several papers that you can physically expand, for example, a cancer biopsy and make those early tiny changes large and obvious. So we had a paper showing that early in breast cancer, at a stage where doctors disagree about the diagnosis, like half the time, right? A coin flip. We can expand those early breast cancer biopsies and train a machine learning algorithm, a simple AI, and it can classify the tumors much more consistently. So there are many preclinical and clinical studies ongoing for both of these technology suites.
Oh, that's really amazing. I wonder if you see a timeline on when they might be approved for wide-scale use. Obviously, we have to do the foundational studies to make sure it's safe and all these other factors. But what do you think about the timeline on that?
Well, Leeway and I started a company called Cognito Therapeutics, which is trying to get the Alzheimer's therapy through the FDA. The European team that's trying to get the blindness treatment, I believe, got FDA fast track designation recently. So I'm not a doctor, I'm not a clinician. I'm not leading these efforts. I just make the stuff and help people use it to make discoveries. But these are all things you can look up on Google News if you want to see more about it. Yeah, I mean, I myself am very excited about the prospects, but I don't know enough about I'm not a doctor or clinician, so I'm not leading these efforts of doing the clinical trials.
Surely, I understand. I definitely have other questions, but I also want to leave some room for others to ask questions. So I'm not sure if it's to Wisdom or Josie. Do either of you have questions?
Yeah, I have some questions. First of all, I just wanted to thank you for spending time with us. I really appreciate this chance to chat with you.
Great.
Um, yeah, I'm a PhD student working in your neck of the woods. I live in Kendall, but I walk the Longfellow, work in a Harvard lab in the MGH campus. And our lab, like many others, really depend on the tools that you've been building. So, just want to thank you for your work in general.
Happy to help.
Um, yeah, so I have two questions. One is more specific to my research, and the other one's a little broader. Um, so my lab and myself, like many neuroscience labs, are keeping a close eye on your work. Your work with improving opsins, for example, step opsins, red shifting, voltage imaging, all of these kinds of tool building projects you have going. I'm in the small camp, but growing, I think, camp, interested in dendritic computation. And I know there's been a lot of good work around being subcellularly specific, somal localized mostly. I'm wondering if you have your eye on, or if you have any thoughts about the idea of building dendritic specific manipulation tools. That would be the first question. And the second one is more broad about, I'm curious what you see on the horizon, what you're excited about, where you think the field should be focused moving forward.
Always happy to help. Yeah, I mean, there have been several papers where people have expressed molecules on dendrites in the past. If they're not up to speed for what you need, it's always good to hear feedback. But yeah, I always like to understand the problem before trying to solve it. Yeah, so I'd love to learn more about what you're struggling with, why the existing tools don't cut it for you, and what we could do. Yeah, so maybe email me. I'd love to learn more. But yeah, you don't know what you don't know, and I have seen several groups that have done dendritic targeting or tried to, stimulate parts of cells by focusing light, and it's just great to know why those things aren't good enough before we spend a lot of time trying to make them better. Your second question was about what's next. Is that right?
Yeah. What are you excited about? What do you think the field should be focusing on?
Those are two different things. I mean, the field kind of has its own will, right? And there's the people have certain priorities, and they perceive needs as a collective in certain ways. What I think what the field seems to want and where I'm going are quite different, which is as it should be, I guess. We don't want to be redundant. I don't want to do things other people could do. Um, yeah. So in terms of but I think we, in a nutshell, just want to see everything in biology. Why can't we see sugars? Why can't we see fats? Why can't we see vitamins? Why can't we control these things? So much of our group is focused on seeing everything and controlling everything in biology. That's really what I see the core problem to be. Now, what the field wants, that's really up to whatever the field wants, I guess. And we definitely try to help people solve problems by providing tools to them freely. And two past tool sets, the optogenetics and the expansion, are used by perhaps thousands of groups now. And so we're always trying to share our tools with everybody who wants to use them.
Awesome, thank you. Yeah, you're always been very generous with sharing the tools you're building. So thank you,
I
and
don't,
I'll take you up on that email.
yeah, I don't see it as generosity. If we work so hard to build these tools and nobody uses it, what's the point? I should just
Hmm.
retire and lie on the beach. Why work so hard if nobody's going to use it?
Excellent, excellent. Thank you for taking my questions.
Yeah, I have a
Have a-
follow-up question about, we've been interested in astrocytes lately, and we had the room where we talked about you actually, about how astrocytes are more involved in transmitting information than we thought before, and this researcher was here from Switzerland who talked about that the potassium signaling from neurons is actually kind of the trigger for then astrocytes to start then their calcium waves. And would there be a possibility in the future to have a good potassium indicator?
Yeah, good question. Christina Torres Caban, who recently finished her PhD in my group, um, worked on an improved potassium indicator. She's now finished up and moved on. Yeah, it's early days, from the first calcium indicator in 1997 to GCamp, the most popular one. That was, what, uh, 97, 2007, like 15 years. It takes time to build great tools, but we and a couple other groups have begun the process.
Yeah, that's wonderful. And you mentioned, sensing everything. So, are you thinking of, developing sensors for humans to basically monitor better for preventive care, maybe even in the future, to sense more and like to collect more information and then be warned in the future more like earlier on so it doesn't develop maybe into a disease, something?
Well, that's what I mentioned earlier. So we and others have shown that the expansion method can be applied to human tissue biopsies, and we can then classify cancers at an earlier stage than we could before. So yes, that's already published work from our group, and we're working with many people to apply expansion to many different areas.
Yeah, thank you so much. And sorry, Josie and Nardish, I know you were waiting patiently. Please go ahead. Thank you.
No, no worries. Can you hear me, first of all?
Perfectly.
Great. Well, Denise kind of had my same question, so I'll be a little bit more specific with it then. I'm really interested in how microplastics influence neural development. I'm wondering if your expansion technique is being applied or could be applied to perhaps examine like how microplastics localize within cells or how they could be influencing development. Thank you.
So the expansion protocols we post on the website expansionmicroscopy.org. And as I mentioned, over 450 experimental results have already been published. The vast majority of these results are from people who I don't know. So the protocols are out there and anybody can apply them. So I think the honest answer to your question is I don't know what people are doing with it. It's out there. We want to help everybody do nanoimaging. But yeah, it could be worth a try. I think you could probably expand cells and hunt down where things are inside of them for sure.
All right, thank you. I'll pass it to Nerdish then. I don't have much more questions.
I'm in a really bad area. I appreciate the talk. Thank you.
Yeah, don't worry about it. So I know, Serena, you said you had more questions, and Wisdom and Frank, do you want to ask your follow-up questions?
Well, sure. So in terms of more sensing, Katerina just mentioned the potassium sensing. Um, voltage sensing has also come up. I wonder if you have your sights on any of that in a dendritic context.
Yeah, so we published a improved voltage indicator in Nature Chemical Biology in 2018 that we found with a robotic screening method we developed. And then a year later, 2019, we had a paper with Hans group at BU in Nature where we showed that we could use this to image neural activity in awake behaving mice. And we and others have used these to look at dendritic voltages. And in fact, a group, Chris Dula's group at Tufts, just published a nice paper in Nature Neuroscience, to go back to an earlier theme about astrocytes as well, showing that they could image voltage on astrocyte processes and see how those relate to neural activity. So absolutely, it works, and the field's always getting better.
So that's fascinating. I've also sort of fallen down the astrocyte rabbit hole. In terms of getting at the calcium oscillations and the drivers for those, and not just the slow wave oscillations, but the limits of the high frequency oscillations in the processes. Um, Anything in your crosshairs for that?
For oscillations?
Calcium oscillations, yeah.
Well, I guess the question I would have is what do the existing tools fail to do? I mean, many people have built calcium indicators. It's a giant industry. People can look at calcium transients and oscillations with them. I guess I would ask what's wrong with those tools? What's the need that's unmet? And I just love to understand the problem before we propose to tackle it.
Fully appreciate that. As I'm wading through the literature, and it's really just coming out at an increased pace. Um, there's there's discussion and debate about just trying to understand the role of these calcium oscillations in what I like to phrase as an astrocyte neuron dialogue, and how much information processing are they carrying, and what their actual role is. In some sense, some reports will indicate they're pushing neural synchrony in topographic regions, which is kind of interesting. But the significance of the specific frequency, are they in some cases the alpha and beta region, beta rhythms, other cases they're much longer. Um, but there is difficulty in getting to the resolution of the finest processes and what they're actually doing, and whether those signals are jumping from astrocyte through the syncytium or propagating in other regions. I guess like in any interesting forefront area, there's so many more questions than answers. And
Yeah. But as a technology inventor, my main question is what's wrong with the old technology? So it sounds like you're saying the spatial resolution is too poor. Is that an accurate summary of your statement?
well, yeah, and getting the in vivo localized spatial resolution to answer questions of whether the activations in this process are driving plasticity changes in other neurons within the domain of the astrocyte, or so both the fine spatial and temporal resolutions, really pushing the limits to what we can capture.
So the next step then is really to define the problem in terms of numbers. What is the number that describes the resolution you need in space, and what is the number that describes the resolution you need in time? Once you have a number, then we can build a tool, right?
Hmm.
But I really need to know the number, and that's the problem. And then once we know what the number is, that's the problem, we can devise a solution. But when it comes to things like resolution in time or space, the number is the goal, right?
Well, so if you're in submicrometer and millisecond regions.
So a confocal microscope should have a lateral resolution of 300 nanometers, and
Hmm.
if you do line scanning, it should be able to go at 1,000 per second, right? So I guess the question is, what's wrong with the existing technology? I really want to understand what's wrong with the existing technology before I can
Hmm.
propose a solution to it.
And I think confocal doesn't quite get to the finest processes of the astrocyte. At least that's what sticks out in my mind. Um, so
So it'd
we're
be
down
great to
to
collect.
hundreds.
Sorry,
Mhm.
go ahead.
So I think we're down to the hundreds of nanometers then.
Yeah.
Go
So
ahead.
go ahead. Sorry, you go 1st.
No, go ahead.
Well, I just think then the right thing to do would be to collect some data with the flawed technology and evaluate why it's wrong, right? That's what I was referring to at the beginning of this little conversation, what I call constructive failure. You have to fail to understand what the problem is, right? But I
Hmm.
don't think I understand what the problem is quite yet.
Was it confocal down to 400 nanometers? You said 300 nanometers.
Well, the textbook would tell you the confocal lateral resolution is 300 nanometers, but then the question is what's wrong when you actually use it, I guess.
Hmm. Okay, thank you.
So I have a question from the audience. If that's okay, like that was in the chat and asked me in the back channel. Um, so the biggest question is: do you think optogenetics will be awarded a Nobel Prize? And do you think, yeah, do you think that this will happen? I would answer yes, but please go ahead. Ed.
Um, I don't know. I mean, it has won several awards already. A few of us received the Gardner Award, for example. Some others received the Lasker Award. There have been some awards awarded for it, so who knows? Maybe, maybe not.
Yeah, Yeah, I agree. It has won a lot of awards, and it has brought us so many insights that we could have never done without it. So, yeah, I think it's very likely. And you know, you develop so many really amazing, interesting tools, also for mapping the architecture in the brain. I think that's also really interesting. Do you think, because you mentioned in the beginning you come, probably not anymore, I don't know, do you think you still have a different bias perspective than people that are, you know, doing the neuroscience part, or do you feel pretty much you were the same bias as we are?
What do you mean by bias?
Well, not by, you said in the beginning that was maybe helpful that you came from a different field and then joined the neuroscience field basically to develop these tools. And I think you kind of said you had a different perspective in the beginning.
Hmm.
Do you have now still, do you think you still have a different perspective from people that came from biology and neuroscience, or did you kind of merge?
Well, you tell me. I mean, my vision is we understand the brain to the point where we can simulate thoughts and feelings on a computer. And that's because we can simulate the molecules throughout cells, all of them, and the cells throughout the brain, all of them. So that's my vision. And I like to do that for some small brains like worms and fish. Um, yeah, I mean, we'd love to collaborate with more people on that, and we're trying to build a network. But yeah, I really just I think of the brain as a system that runs on chemistry according to physical rules that implements computation. So that's how I think about it. Whether other people think that way or not, I couldn't tell you.
And so, do you think we will know enough to simulate it? And will we know enough? I don't know. When do you think we will know enough to, let's say, to simulate a person's brain? It's probably something you got asked, and it's probably impossible to answer, but it's still an interesting question. So, thank you.
Well, this is what I like to call real science, right? It's not like a cookbook where you follow a recipe and at noontime you're guaranteed lunch, right? This is real science where, we don't know what's going to happen next. So I can't answer your question.
Yeah, that is a real scientific answer. I agree. We cannot possibly know that, but so in very simple organisms, you mentioned the worms. You know, we know all the neurons, and we know a lot of molecules involved, and so on. Um, do you think that's enough for us? Like, could we have a model, a full model of a worm by now, to have like a full simulation um, run based on, because that's probably, the worm is probably the one we know most about.
I don't think anybody's mapped the molecules, connections, and activity of a single worm, right? Um, I think that's what we need. Suppose I record the activity of my brain and we get the connections of a second person's brain and we get the molecules of a third person's brain. I don't think we could add them up, right? Any more than taking the keyboard from a PC and the screen of an iPhone and the computer of an Android phone and mashing it all together would result in a functioning device. So what we want to do is to look at the activity and the wiring and the molecules all of the very same brain. And I think that's required. And I don't think that's been done before.
Yeah. So do you think we really need to know every single molecule to know how thoughts work, and you know how behavior works in general? Because you know, it's very different when you talk with people from different fields. Mostly if I talk with people that come from AI fields, they would say, like, a lot of them would say probably no, you don't need all this information, you probably just need the surface of the information, and then you have enough to simulate something. So do you have a view on that, or is that also, you know, not something you would like to, elaborate about?
Well, if somebody can solve the brain with existing knowledge, please do it, and then I can retire and go lie on the beach. That'd be great. But until that happens, I'm going to keep getting down to the ground truth, the fundamental building blocks of life.
Yeah, I agree on that. I just wanted to.
But absolutely, if somebody can solve the brain and we can understand thoughts and feelings, I will gladly retire. If we fully understand the human mind, then we're done.
Yeah, I agree, but it's
But I haven't seen that yet,
yeah, yeah, I totally.
so I will continue to work.
Yes, and what I think the question was asked before, what you're working on right now, what is happening in your lab right now in your world?
Well, I think we summarized it earlier. I'd like to see all the activity in a brain, map the molecules and connections of that very same brain, and integrate that all into a model of what decisions, emotions, and so forth are.
Yeah, I think that's really interesting. Do you think, or could you maybe see that they are not just different types of neurons, but within the neurons, that we will come up with a better classification of synapses? Do you think that's something we will have to do better?
I guess my question would be, why do we need to classify the synapses? Would it be enough just to describe what they do, or I don't know. I don't feel the need to classify. I would rather just understand the mechanism of how they work.
Yeah, yeah, I can see that. Right now we do like mushroom types. Like I don't know if you work with synapse people.
All the time.
Sometimes they do, right? They
We
they
just
give these
published a
classifications, but I'm not sure how. I agree. I'm not sure how useful they are, but yeah, we kind of attempt that, but
Well, they're what I
yeah,
call human-defined words, right? We code names. Like even the word neuroscience implies that we study neurons, but what about glial cells, right? I mean, there are all sorts of other cells. So I'm always very skeptical of human-defined words. And so what I really want to do is what I said earlier, to understand the brain as a computer science system that runs on chemicals according to the laws of physics. And that's really how I want to think about things.
yeah. Do you think the neuromorphic field is really exciting and interesting in that way? Um,
I don't know much about it. No, I don't know much about it. It could be amazing, but I just don't know much about it.
Yeah, it's kind of basically attempt to do what you're saying, to make like artificial synapses. Some use real chemical gradients and so on, and or just simulating them in a way. It's a really interesting new field, but I think again the better communication between what actually data is out there from neuroscientists, and then communicating them well with engineers that are working on that is really very helpful. So I'm so impressed that you did that, and you do that. You keep doing that so well. So what's how do you do that in everyday life to keep this communication up very well? And could you give advice for for the field to be more multidisciplinary? Should people stay broader, like kind of you did in your education, to be able to switch fields? Should people do that more? Like, what would your advice be for maybe developing better AI and models?
Well, I don't know much about developing better AI, so I can't help you on that question. Regarding fields, I try not to think about fields very much, because again, those are human-defined words, right? Why should we stop learning just because now we've gone from physics into chemistry or chemistry into physics? I just don't see the boundaries between them very well.
Oh, I love that answer because That's why we did the science society and bring people from very different fields together. We kind of see it very similarly. So that's really amazing. Um, so I think should schools change their program? Like, should there be a general science degree and not, bachelors of different types of science fields?
I'm not sure. I mean, I was an undergraduate for 6 years, right? I think the world's gotten much more fast-paced since then. I did college before there was a department of biological engineering at MIT or of bioengineering at Stanford. That's all pretty new. So I spent 2 years learning chemistry and then 4 years learning electrical engineering, computer science, and physics. I don't know anybody else who spent 6 years as an undergrad since then. So, um, yeah, the world's gotten so fast-paced. I don't know if the world has patience for such things.
Yeah, that's true. But in general, like, don't you think in developed countries, youth, like the time we have to be spent in youth keeps increasing? Like, for example, you were able to be on your parents' health insurance until 25. Back in the days, maybe it was until 18. Like, for example, I grew up in Germany. I'm from Portugal, but I grew up in Germany. So there you could drop off school with 16, and now you have to stay in school until you're 18. I feel like we have that sense, but in reality, government and legislations and all of these give us actually more time to spend in this youth state. So maybe it's a money thing in the US that it's also very expensive to stay longer. I don't know. Do you think there's like a cultural difference in that?
There are a lot of differences. I mean, I think there is also the question of privilege. If somebody does have wealthy parents who can put their kids on their health insurance or whatnot, maybe they can go to school longer. But I know a lot of people who had to go work because they started having children or they needed to have a job to make money. Um, and so I think, a lot of questions about equity in educational systems as well. Yeah, this is a complex societal topic that touches upon every aspect of society, I'm afraid.
Yeah, I agree on that. And yeah, I was a very young mother with 18, but gladly I grew up in Germany, so could go to
Mm,
college for free. I even got money every month if I had good
wow.
grades. So yeah, it's very different.
Yeah.
But if I wouldn't have been there, I wouldn't be here now talking with
Oh,
you. So thanks,
amazing, amazing.
Brittany. And
Yeah,
Victoria
thank
has
you.
a
I'd love to
question.
just jump in. What you asked, Katarina, was similar on my mind. I was curious. So I work in arts education, but integrating arts into science. And so when I'm hearing you talk about, that this word, this is, human derived from our language and it shouldn't dictate our science, and also understanding that that life is all interrelated, so that it's very artificial to regiment subjects even, but I recognize that we do that for convenience sake even. But my question is, if you could change something, or if there was a certain path of education that you think, maybe a philosophy that you would follow,
Hmm.
what would that be? Just for example, I feel like it would be great to expose kids to something like calculus way before arithmetic, just because it's conceptual and kids love concepts. They don't need to be able to do things necessarily correctly, but kids are so concept driven. So, yeah, so that's my question.
I think individuals are so different from each other. I mean, I have two children now, and they're quite different. And then I feel like my mind is actually quite different from how a lot of people deal with science. I mean, I think my mind is very language oriented, and I think of scientific concepts almost like stories. Um, so I'm not sure there's a one size fits all in terms of even how to teach mathematics. Um, my two children learn math in, I think, very different ways. Um, yeah, I don't know much about education. I think I've taught college students and graduate students at a certain point where they're really quite advanced. But But I think early in life, people learn so differently. When I started learning biology, having trained in physics, I was overwhelmed by all the acronyms and alphabet soup. There are 30,000 genes in the genome. Do we have to learn all 30,000? And all the millions of variants? And so I started treating biology like a language. And I almost feel like I trained my brain a little bit like an AI, here are all these different relationships between concepts, and then I would extract patterns from them. So I think of biology quite differently than somebody who kind of grew up, learning biology. But when I started graduate school in neuroscience, I hadn't taken a biology class since high school, so I knew so little about it. So yeah, I think it really depends on how individual minds work, and I
Yeah,
think we
well,
know so little
yeah,
about
sorry.
that. Go ahead. Mhm.
No, that's why I asked you, because really it isn't in my thinking, it's not the science of education that would be the best at directing education, because everyone is different, but it's really people who remember how they actually learned, not how they were able to prepare for a test, but how learning happened and how engaging with a topic happened. So you're describing, creating or noticing, recognizing patterns of concepts and how you worked. So that's what I think is really exciting when people, maybe you can apply that kind of compassion to your students when you're sharing information with them, that they might be learning it so many different ways, and allowing for that. That's a great mistake I think that we make, is not recognizing what you said, that everyone's an individual.
Yeah, I mentored 250 people in my group, and who knows how many hundreds more in my classes. Every person is different. And I think partly why I customize mentorship to each person, some people might need to collaborate more. Some people should collaborate less and read more. Some people might read too much and should get out and play around. Some people are too playful and should be more disciplined. Everybody needs different mentorship. And I think that's one reason why our alumni have been so successful and gone on to start climate change companies and 3D printing companies and going after it, revolutionizing education and trying to, you know, start institutes right out of graduate school and so forth, is because they have all learned in their own way. It's so key. People always ask me, "What's your mentorship style?" And I'm like, I don't know how to answer that question. If you ask 5 people in my group what my mentorship style is, you might get 5 opposite answers.
You remind me of, there's an educator, Paulo Freire, and Catarina, I think he was Portuguese, forgive me if I mispronounce his name, but exactly what you're describing is that he said he fought back against, well, he wrote his major work was Pedagogy of the Oppressed, and he fought back against what he called the banking system of education, where teachers would consider that students were vessels to be filled with information. Instead, he asked that we recognize that everybody comes with their own experience and bank of information, and that's what we need to see, so that we can help everybody, just not allow, but just get out of people's way and facilitate whatever the heck they need, because we all come with different treasure to the classroom.
Yeah. The one thing I would push back a little bit is what people need might not be what they want always. And I sometimes feel like I have to be the opposite of the student, right? If somebody is way too collaborative, I might need to get them to focus. But if somebody's way too focused, I might need to get them to collaborate a bit. And sometimes I feel like my job is to sort of, be the dialectical fusion. And yeah, I haven't read this book, so maybe I'm being redundant with it.
Not at all.
But uh, but I do think that just giving people what they think they want might not be what they actually
Yeah,
want when it comes to education, anyway.
right. Yes, of course it is. There's a lot, but that's definitely worth mentioning. So I see Dr. Haida is here, and would you like to join?
Thank you, Victoria. Yes, yeah, it's a beautiful discussion. I'm a big fan, actually, of neurobiology, and I use it in my teaching in tertiary education and secondary education. So, to give a context, I'm actually based in Australia, in Melbourne, and working with a few universities, Melbourne Uni, Flinders Uni, and South Australia University, for STEAM education. So, big fan of utilizing the left and right brain. So thank you, Dr. Boyden, and thank you, Victoria. This is Heidi, and I'm complete. Thank you.
Wonderful meeting you.
Likewise, same here.
Thank you for joining our stage. Go ahead, Katarina.
Yeah, I wanted to check in with Ed. I've not been going for an hour more or less, so wanted to check in, you know, give you an opportunity to get rid of us, or if you have a few more minutes, please let us know.
Yeah, probably 4 minutes. My kids are home now, and they probably want to have dinner at some point. But if there are other questions, happy to answer them.
Yes, Serena, go ahead.
So I was really curious about a follow-up on some of the ideas you expressed in terms of how far we can go in simulating the detail of life. It's been a couple decades on the large-scale simulation, both following, the advances in resources and how much further we can reach, as well as, being perilously under-resourced in terms of the, full picture of what nature serves up. Recently, though, I got involved in just a separate side project, looking at, how we might take it a little step further in terms of subcellular simulation and phenomena. And I'm wondering, in terms of your thinking about approaches to the level of fidelity and the detail of what you would want in a simulation for, in part of the motivation behind the question is, in I did want the aspects of chemical diffusion present, but not in atomistic, full detail of empirical force fields, all atom force fields, and so forth. What it led me down to is, well, what we really need within the simulation is some type of developmental protocol to get us to, embracing extremely complex simulations. It's very difficult to set them up. But if we can draw on principles of multi-cellular systems and how they develop, perhaps we could grow more complex arrangements. But it's always tricky where you cut the abstractions. Just wondering on your thoughts on next stages in that type of detailed simulation that you expressed interest in.
Well, whether you're simulating development or the mature state, you still have to simulate according to some rules, right? I think that what we want to do is to map out what's actually there and see what the interactions look like, and from that derive the rules. I'm very empirical.
Mm-hmm.
So that would apply to whether you want to simulate development or the mature state.
Well, even in terms of mapping out what's there and what you begin to simulate, um, I'm assuming you're not going down to atomistic detail, or are you?
Well, I don't know. This is what I like to call real science. We don't know when to stop, right? I'm not going to give anybody a cheap approximation here. I'm going to tell it like it is. And anybody who tells you they know when to stop and they can guarantee a simulation life is probably wrong or lying or both.
So in terms of the next stages, though, we're certainly not ready for a fully quantum mechanical treatment of uh, an entire brain. For
Do we
example,
need quantum mechanics though when we're at room
I,
temperature or body temperature?
I personally don't think so. It's an interesting discussion, and um, I don't think we're done with Maxwell's or anything. But it's an interesting discussion whether there are any quantum mechanical effects that are relevant to thought and behavior.
Yeah.
Um,
Well, extraordinary claims do need extraordinary evidence. There have been several analyses of how long you can keep coherent states in a messy body temperature environment like the brain, and it seems unlikely. But if somebody can prove it,
mhm,
I'm driven by the data. What matters is the data in the end.
mhm. Are you? I guess I should ask: are you actively pursuing more detailed simulations?
Right now, we're focused on the mapping. We want to know what's there and
Hmm.
how it interacts. Without the data, I don't think we're ready to simulate.
I think.
Excellent.
Oh, thank you. I think we skipped Ilya, and I wanted to acknowledge Ilya that he's here. And I'm sorry. Did you have a question?
Yes, I do. I don't know if can you hear me? I'm on Bluetooth.
Perfectly.
Okay, awesome. Hey, so I apologize. I joined a little bit late, well, quite late. And I missed some parts, but I heard some other parts, and I'm curious. So tell me if it's feasible or not, or if I'm thinking it correctly. When it comes to gene therapies, is whatever tools you've designed, is that something that could be used?
Yeah. So optogenetics is, as I mentioned earlier, being used in Europe in clinical trials for blindness, and that's a gene therapy using standard gene therapy vectors. That's not my area of expertise, but there are many people in Europe who are doing that kind of work.
Yeah, I think it's really impressive that they started this because people tended to say, oh, this will still take a long time, but they have really impressive results, so it's very promising.
Yeah, please, Ilya. I just temporarily muted because it sounded like there was some sound coming from your mic and we couldn't hear Ed's answer, but I didn't want to imply that we were stopping you in your path to questioning.
Yeah, I realized that. I'm sorry. And so I guess the second part of the same question. So that's, excuse me, that's optogenetics, right? So that's I'm guessing it's a visual thing that requires some inputs for that to work. When it comes to like more neuronal diseases, right? This is what I'm after, truly. Does the same concept applies?
Does what concept apply? I didn't quite follow.
So like you said, they're doing trials for vision related diseases. What if it's not vision related disease?
Nobody's pursuing that yet,
I see.
so I don't have
All
an
right.
answer to that question.
Thank you.
And LT, hi. I think one of the last questions will be yours. Please go ahead.
Thank you, Katerina, and thank you, Ed. And my question is related to because the other disease you mentioned is Alzheimer's disease, right? Is it carried on
Correct.
in US or it's in Europe?
That's in the United States, yeah. So Li Wei Sai is an MIT professor. She used optogenetics to discover a brainwave that in mice could clean up Alzheimer's. The teams then went on under Li Wei's leadership to develop movies, flickering lights, and clicking sounds that could also clean up Alzheimer's in mice. And now, both in a company that we co-founded to do scalable clinical trials as well as in multiple university trials, there are several teams now that are trying to see if movies can indeed help clean up Alzheimer's disease.
So clean up a plaque in that case is, may I ask, for in the mice?
Not just plaques, but hyperphosphorylated tau, microglial
Okay,
inflammation,
okay.
improving blood flow, and the list goes on and on.
Wonderful, wonderful. Thank you so much. I have to look up because I'm really interested. Thank you. It's a wonderful discussion.
Yeah,
Thank you,
yeah.
everyone. Yeah.
If I could follow up on this. So yeah, this is what I was curious about. Is it just limited to Alzheimer's, or is that the model you chose for your proof of concept?
Well, Li Wei Sai is an expert in Alzheimer's disease. So her group led the project. Our group is helping from the technology side. But now that the papers have been published, people have grown interested in applying this way of thinking to many different conditions. Absolutely. Yeah.
If I may, Ilya, ask a question, and I think related to that, Ilya, you can find on the website we shared maybe checking out also the optogenetics hardware at you developed, which is also really impressive for in vivo manipulation that is very precise in a 3D space, combining it with the holographic photo activation. Just click on optogenetics hardware enabling neural control by light. It's really impressive, and I think it will. But Ed, maybe you want to add something to that
Well, first of all,
information.
it's really important to point out who led the work. So Valentina Emiliani at the Institut de la Vision in Paris led the work to build the holographic projectors. That's not our work, but it's work led by her. But yeah, we did collaborate with her to build holographic strategies for controlling single neurons in the brain in 3D.
Where would I see this? I'm looking for it right now. Is that syntheticneurobiology.org?
Yeah, so if you click on the link that's on top, and then you go to a project, I believe, and then there are the different projects that the group is currently working on, and one is the hardware enabling neural control by light. So it says that, I can just read that Ed and his colleagues have developed a set of optogenetic reagents that fully encoded reagents that when targeted specific cells, that's kind of optogenetics. You can control them via light, and then you have these different light transmitting electrodes or cables, wires, and you can then precisely in a 3D space control in a living animal, which is really important because it's really interesting to see what different cells in the 3D space do in a behaving animal. I don't know if you want to add something to that, Ed.
Yeah, happy to answer any questions.
So other than not having to have a 2-photon microscope set up, is there any spatial temporal advantages to this device over doing, say, holographic stimulation with an SLM or something like that?
That's what it is, yeah. So Valentina pioneered that. She pioneered 2-photon SLM holography for neural control. That's her invention.
I see. I thought we were talking about a implantable device or something. I'll look at the link.
Oh, no, this is not implantable yet. No.
Gotcha. I'm with you.
Well, on the website you have one implantable device, like that has different cable, like optic cables in there, and then I believe you can stimulate them. Is it a string of LEDs, and that permits you to enable blue light and red light and different heights, basically, of that string, or
So let me propose a way of thinking about this, because otherwise we're going to get bogged down in details. Light sources are a dime a dozen. In your computer, in your phone, on your ceiling, there are LEDs, lasers. Light sources are dirt cheap and ubiquitous. So you can take those light sources and arrange them in any 3D pattern you want, right? That's what the consumer electronics industry has done for you. So so I wouldn't focus on any one light source or device. Instead, I would just say, look at your cell phone, look at your automobile, look at the light bulbs on your ceiling, look at, you can make light sources in any 3D geometry you want nowadays. So if you want to make it in some 3D shape, you can do whatever you want. That's how I think about it. So all these examples we're talking about, we're kind of getting bogged down in details, but they're the way to think about it is just to think that light sources are everywhere, right? And you can make light sources any way you want. That's how I think about it.
yeah, thank you. Yeah, that's a really great way of explaining that. And did anyone have any last questions or comments?
Yes,
Please go
Katerina.
ahead.
Thank you.
Oh, okay,
Yes,
go ahead.
and excuse the noise behind me because I'm in holidays. I'm so sorry. Just a quick question about epigenetics. It may be related to Elia's question. The epigenetics and biomimicry. The advances in using both of those sciences in neurobiology. Are we getting there or not yet? Thank you.
So I'm afraid I don't know much about those topics. I'm more of an inventor who makes technologies to see and control things, but I mean, certainly you can apply our tools to anything and everything. So yeah, if you're interested in those topics and want our help with our technologies, happy to help. But epigenetics, I mean, people are definitely applying our tools to that. Biomimicry, you could study all sorts of biology, but I definitely am not an expert on those topics.
We start an early research in Melbourne Uni using the beetles and the camouflage and photonics as well. So we may discuss it further on another arena or academia, because I really admire what Katrina trying to do here, uplifting the level of Clubhouse to be a very sciencey, academically placed. So thanks to Katrina and thanks to you to be here with us today and have this deep discussion in science. Thank you so much.
Always happy to help.
Yeah, thank you, Heidi, for saying that. And yeah, I couldn't agree more. It's such an honor that you came and took the time to answer our questions and also have this broader discussions with us. It was such an interesting discussion, and I hope you enjoyed it. And yeah, you're always invited back, of course. I know you're very, very busy, but maybe next year one day we can have you back and enjoy dinner with your children. I know children that are hungry are not easy to control, so
Oh,
we
I
don't want.
know. I'm amazed I'm still alive.
Yeah, so yeah, thank you so much again. And yeah, we are really all happy that you spent some time with us here today. And
And I.
yeah, we are looking forward to follow your work. So thank you.
Stay in touch and have a great night.
Yeah, and thank you everyone for coming, asking questions, being involved. We always appreciate that. Follow the club if you like discussions like this, and then you can see the calendar. And yeah, enjoy your day, night, wherever you are. And thank you again, everyone. And of course, special thanks to you. And
Wonderful talking to you.
Yeah, thank you, Dr. Brodin, to share your precious thoughts with us, and thanks, Katerina. As always, this is always, very, very, exciting times to spend with all the best friends of society here. Thank you to all.
Thank you, everyone, for those running the room. Thank you, Dr. Boyden, for joining us. I'll be firing you an email about dendrites in the future. So thanks for joining.
Have a great day.
okay, I'll close the room in 3, 2, 1. Bye, everyone. Thank you.
See you later.
Everyone.